Hi. Good evening, everyone. Welcome back to my channel. So, this live, I will explain the gene expression part 3. This is the last part lah for Stpm Biology Semester 3, Chapter 4, Chapter 17, Inheritance and Genetic Control. Okay, so this part I will explain about the gene expression, protein synthesis, about the translation. So for the last live, I already explained transcription process. So gene expression process is the process where DNA change will produce mRNA, and then mRNA will produce protein, right? So DNA to mRNA is transcription process. I already explained for last live for the live tadi just now. I just explained in 6 o'clock one. So if you want to listen, you can refer back. This one I already explained. So for this live, I will explain translation process. Okay. So what is translation? Translation is actually, it is the synthesis of polypeptide using the information of mRNA. So it is the synthesis of polypeptide. So polypeptide chain, it is the protein, right? Protein using the information from mRNA. Okay. Okay, so before that, translation process will involve the organelles called ribosome. Okay, I think this organelles everyone familiar with it, right? In Sem 1 already learned. So one of the function of ribosome that Sem 1 you learn is to synthesis protein with the help of tRNA. Dia nak bekerja dengan tRNA for the translation process to synthesis protein, right? This is the function of the ribosome. And also, you also learn the structure, right? But that structure is not too detailed. You just learn at that structure. Normally, in the books, you only learn ribosome contains large unit and small subunit, right? Ada large subunit and small subunit. And also, you learn 70S ribosome and also 80S ribosome. 70S is for prokaryote, 80S is for eukaryote. Right, this is the type of ribosome you learn. So actually it is same. So for translation process, the function actually ribosome is same, but just the types the ribosome are not same. So prokaryote is for 70S and eukaryote is for 80S. Okay, so for the structure of ribosome, you look this structure lah. So the main structure is this. Awak tengok ada lubang-lubang macam ni punya. So there are known as A site, P site, and E site. So it is located at the last unit of the ribosome. Ini site ini adalah dekat untuk last unit. Right? The small subunit is the have contain mRNA binding site. So mRNA will bind at the small subunit, bukan last unit. Last unit is for tRNA. Last unit is responsibility for tRNA. What? Small subunit is for mRNA. Okay, so A site, so this A site, what is that? Does this site mean? This site is the binding site for amino acid tRNA. Means that this tRNA that carry the amino acid. tRNA we know that tRNA dia macam ni kan. Inilah structure tRNA. So dia look like this. Dia macam ni. Lepas tu dia ada got anticodon, right? Got 3 anticodon at here. Lepas tu it carry 1 amino acid. This is the AA amino acid. So this is we call as amino acid tRNA. So this is tRNA. And it is bind with the amino acid. So this one is called as amino acid tRNA binding site. Though this kind of tRNA will bind to the A site. So P site is the bacterial tRNA binding site. Means that it also look like this, but it contain peptide chain. Dia ada banyak peptide chain lah. Banyak peptide chain di sini. it contains many amino acid. It is the peptide chain one. Yang ni hanya for this amino acid only have one amino acid. But for this particular tRNA binding site, it is used for this tRNA. Ada banyak, it has the peptide chain on tRNA. Acid site is for place where this tRNA will be released. It is to releasing the tRNA one. Okay, so this is the general structure of the ribosome, ribosome that play a role in translation one. So awak kena ingat lah. So there are three sites: A site, P site, and E site. And also for small unit, it contain mRNA binding site also. Okay. So next, the first step of the translation is we need to activate tRNA first. Okay, because our tRNA it does not contain amino acid one. Our tRNA, pure tRNA will remain like this. Hanya dia macam ni saja, macam ni saja. This is the tRNA. Dia hanya macam ni saja. This is pure tRNA. And contain anticodon. There is an anticodon here. Hanya macam ni saja. And it does not carry any amino acid. So for this tRNA, we have to activate them. Activate them to carry the amino acid. So this one, the activated form of the tRNA, we call as amino acid tRNA. So this kind of tRNA is called amino acid tRNA. So this is the activated form of tRNA. So how to form this? So these are the steps. You see this picture? These are the steps. So step 1 is the amino acid and the associated tRNA will enter the active site of the specific synthase. So if you see that our tRNA contain anticodon. So each of these anticodon is specified for the amino acid. For the amino acid. So means that they will have total 20 types of tRNA because each type of tRNA have their specific anticodon site. So each specific anticodon site will bind with one type of amino acid only. Like this example, is that the anticodon is AUA. So AUA is responsibility for the amino acid of tyrosine. This is the anticodon. So the complementary site which is codon is AU. This is the codon one, and codon means mRNA. So means that this amino acid, this is the anticodon that is responsible to bind, will carry the amino acid called tyrosine. Dia hanya akan ambil tyrosine saja. Dia tak akan ambil amino acid yang lain. For this AUA, if the anticodon is AUA, it will only take tyrosine. Okay, so how it does it? So got an enzyme will catalyze this kind of reaction. The enzyme will called as amino acid tRNA synthetase. This enzyme, amino acid tRNA synthetase, will play the major role enzyme to catalyze the reaction between tRNA and also the respective amino acid. So if you see this example, it uses tyrosine tRNA synthetase. So if you know the amino acid, it will directly change to the amino acid terms. But if you normally, our general term is amino acid tRNA synthetase. This one tyrosine because of they use the tyrosine as example. So normally in essay question, you don't need to write this one. Don't need to write this tyrosine one. You just directly use amino acid tRNA synthetase. So when they catalyze the reaction, they will require ATP, and then the synthetase will catalyze the covalent bonding lah. It will catalyze the bonding reaction because the amino acid need to bond with the tRNA. So the tRNA will charge with the amino acid is released by synthetase. So finally, it already formed the activated amino acid tRNA. So this tRNA will carry the amino acid called tyrosine because the anticodon is AUA. So in human, normally they will total have 20 types of tRNA. Must have 20 types of tRNA because we have 20 types of amino acid. Each type of tRNA were responsible to each type of amino acid. Okay, so this is the activation of tRNA. So this is the where places macam mana if you see the protein synthesis, inilah protein lepas tu amino acid later they will combine together to form one polypeptide chain. So this is the first process of activation of tRNA. This is the first process. Okay, the next process, translation, actually is the band process lah. It actually same as transcription. It also has initiation and also elongation and termination. Okay, initiation is the place if you see that translation definition is synthesis of polypeptide using mRNA, right? So mRNA, you see that mRNA actually is also long chain. It is a single stranded chain. So macam mana dia need to start the translation? Where do we need to start the translation? Mana? It has a clue. So we start the translation, start the start codon. So our start codon is AUG. And for anticodon will be the complementary AUG, so UAC. So start codon is for mRNA1. This is the sequence for mRNA. Anticodon is for tRNA. And start codon AUG is coded with foo. Is coded with methionine. So in amino acid in protein synthesis polypeptide translation, the first polypeptide chain the first amino acid must be methionine because methionine is coded with start codon. Dia akan start kalau ada start codon. So maybe start codon is here. So this is the initiation process. So elongation is what? Elongation is the mRNA. mRNA will be translated. It will be translated by using tRNA. tRNA akan translate sampai sini. So this is the elongation process. Akan translate lah. mRNA macam bagi dia simbol kan. Lepas tu tRNA will give amino acid. Dia akan provide amino acid based on the mRNA information and then it will combine to form one polypeptide chain until it reach termination. So termination is a place to stop the translation process. And then the polypeptide chain will be formed. So inilah dia macam sama dengan transcription saja. The definition. So initiation is to determine the position where to start. Elongation is to process of synthesis synthesis of the polypeptide chain, and finally termination is the place to stop the polypeptide formation and release the polypeptide for mRNA and also ribosome. Okay, so this is the general overview of the translation. So initiation process. I already say our start codon is AUG. This is for mRNA. mRNA. So the tRNA will be tRNA macam mana? Our initial our start anticodon will be siapa? UAC. So UAC will be the initial of the anticodon. So means that the tRNA with the UAC will be firstly bind to the firstly come to the mRNA first. And then this anticodon, this tRNA will carry methionine. It will carry methionine. Dia akan bawa methionine. So the mRNA, firstly, it will bind to the small unit of mRNA first through the mRNA binding site. After it bind, and then it will copy. Ya, dia akan bergerak sampai dia ada stop codon at the ribosome. And it already have the start codon, the tRNA will bind the tRNA that have the UAC TNT codon and carry methionine, activator tRNA that carry methionine will bind to the will come to the mRNA and begin the bonding lah. The bonding will occur between the start codon with the start codon. So mRNA binding site on this subunit recognize a specific nucleotide sequence of the mRNA just upstream the start process, start codon. Okay, and initiator tRNA with anticodon UAC base pair with start codon. The tRNA carry the amino acid methionine, so it will bind to there. Okay. Okay. Okay, so firstly, our small subunit will bind to mRNA, and then after mRNA is bind, the activated amino acid tRNA that carry methionine will be also come to find the start codon AUG. Dia akan start codon, dia akan locate at there, bind to there. Okay, lepas tu it will require UTP, and then the last ribosomal unit will bind to it. Dia akan mari lah. Lepas tu will bind with the small unit and they form like this lah. Dia mRNA kat tengah-tengah ni lah macam printer kan. Awak pernah tengok printer tak? mRNA ini macam kertas lah. mRNA macam kertas kan. Yang macam kertas lah. Lepas tu yang ni ribosome macam printer lah. And tRNA macam ink. Yang macam printer punya ink lah. Macam tu lah. Okay, so the arrival of the last ribosome unit will complete the initiation complex called initiation factor. Initiation factor are required to bring all the translation component together. So translation hydrolysis of GTP, then you will use UTP lah. So initiator tRNA is in the P site. So if you see that after the last ribosome unit is by our initial activated tRNA that carry methionine will locate at the P site. Directly locate at the P site. Dia tak akan dekat A site punya. Awak tengok. Dia tak dekat A site. It directly located in the P site. And A site is for ready for other types of tRNA, which will be discussed in the elongation process. So after the last ribosome unit is attached, and the initial activated tRNA already bind to the P site, then the initiation process is done. Already done. Okay, next I will proceed to elongation process. So elongation process involves 3 main steps lah. So firstly, for P site, if you to know, P site is for peptidyl tRNA binding site. So P site will contain the peptide chain. So yang ni circle shape ini adalah for methionine. Yang first one only for yang first one ni akan ialah methionine. Lepas tu dia akan ada banyak amino acid tambah sendiri tambah ke sini. So methionine will be located here. So for this diagram, the methionine will adhere. Inilah akan methionine. This is the methionine one. Yang lain semua amino acid lain. So for the first step is codon vagonization. So yang ni means that means that yang ni, the first step is yang tRNA dekat P site dia tak bergerak. Dia datang ke sini saja. Dia tak bergerak. tRNA dia tak bergerak. Lepas tu the A site will have these 3 codon, right? If you see these 3 codon. So the A site will based on the codon of the based on the codon information from mRNA, the respectively anticodon will be bind to the A site. Maksudnya kalau A site punya information for codon is let's say CCC. Let's say CCC. So our anticodon will be GGG. Okay, so that means that GGG means that the activated the tRNA activated tRNA that carry anticodon GGG with amino acid will be bind to the A site. It require A site. Akan bind dekat A site. Bind dekat. So you will see that the anticodon of an incoming, so it's a incoming amino acid tRNA base pair with the complementary mRNA codon in the A site. So maksudnya yang ni anticodon it must be complementary to the mRNA lah. Maksud macam ni lah. So kalau codon is CCC, so it must be complementary to the anticodon one. So only this tRNA that carry GGG anticodon with their respectively amino acid will bind to the A site. Yang lain tak boleh. Yang lain semua tak boleh. So this is the meaning lah, the first sentence's meaning. So this process also require GTP. Kerana dia kena GTP pula. Okay, lepas tu after the amino acid tRNA activated form is bind to the A site, it will proceed to the second step. Second step is peptide bond formation. Maksudnya peptide bond formation. Maksudnya dia kena form peptide bond lah between the amino acid of A site and P site. Maksud dia macam ni. So the peptide chain from the P site will be breakdown. Yang ni akan breakdown for that it is bonded with tRNA, right, for the P site. Dia akan cut down and then bond with the amino acid bond with the amino acid that at the A site one. Maksudnya dia akan pergi ke sini lah. Yang peptide chain ni akan pergi ke A site one. So the RNA so this process is require RNA lah. RNA molecule of the large ribosomal unit will catalyze the formation of peptide bond between the carboxyl end of the growing polypeptide in the P site. So this one is carboxyl end. Yang ni if you see that this one at the bottom one, this is the amino end. This one is carboxyl end. So if you know the basic unit of amino acid, then you will know what I said. Basic of amino acid contain amino group and carboxyl group, but yang bind dekat tRNA one is end with carboxyl. Okay, so this step remove the polypeptide from the tRNA in P site and attach it. So yang ni senang cerita lah. So yang ni akan remove lah. The peptide chain at the P site will be removed from tRNA from P site and then will attach to the amino acid that attach to A site one. In the A site one. So maksudnya lepas tu dia akan jadi macam ni lah. Tengok dia akan jadi macam ni lah. So protein peptide chain dekat P site ni semua pindah kepada A site ya. So P site sekarang dia tak ada protein. Boleh tengok P site dia tak ada protein. P site hanya ada tRNA sahaja alone. It is not activated form. Okay, lepas tu dia ada translocation process, the finally process of the elongation. So the ribosome, yang paling penting adalah first sentence. The ribosome will translocate the tRNA in the A site to the P site. So maksudnya yang A site ni, the tRNA at the A site will be translocate to the P site. Dia akan berpindah dari A ke P lah. So, yang macam ni lah. A dari ke P. So, yang P ni, if you see, so yang P ni, yang original P ni, dia tak ada guna lah. Sudah tak ada guna lah. So, dia akan pergi ke E site. So, firstly, P ni dia kena pergi E. So, barulah A ni boleh pergi P kan. So, this is the simultaneous process lah. Dia akan berlaku bersama saja. P masuk ke E, lepas tu A akan masuk ke P. And then, yang tRNA at the E site one, actually dia tak ada berguna lah. So, E site is what? E site is acid site. So, acid means what? Dia keluar je lah. Lepas tu ke dalam masuk dalam E site, lepas tu dia terus keluar lah. Yang ni tRNA dia akan keluar daripada ribosome lah. And then maybe it will go another type of activation process lah for another type of amino acid. So this process is repeated. Dia macam repeat saja. It is repeated and repeated again until it reach the elongation. So after that, another type of activated. So lepas tu mRNA ni akan bergerak. So tadi yang 3 ni kan, sekarang dia akan guna yang 3 ni kan. 3 ni. Lepas tu dia akan guna yang 3 ni. Lepas tu dia akan guna 3 ni. Macam ni lah. So mRNA will move from this side until it translate it reach the termination process. So ini process is it is the cycle process repeated until it reach the termination region. Kalau dia tak ada termination region, the process is repeated only. Dia akan produce the polypeptide chain until dia sampai termination. Okay, so next is the final step is the termination process. It is the stop process of the addition of polypeptide amino acid to stop the formation of polypeptide chains lah. Okay, so termination is kalau lah stop the A site of this, the mRNA baca mereka sudah bergerak sampailah dia the codon is stop codon. So either 3 of these, UAG, UAA, UGA. This one awak kena hafal. Memang kena hafal. Yang ni awak memang kena hafal. So in the genetic code table, actually in genetic code table, you don't need to memorize one. But if you can memorize, it's good lah. But for the amino acid, respectively coding, actually don't need to memorize one. But there are 4 code you have to memorize. Only 4 code. First code is AUG. This is the start codon. Awak memang kena memorize. And it is coded with methionine. And another 3 code is the stop codon. Stop codon memang kena memorize. UAG, UAA, and also UGA. These are the stop codon. Kalau lah mRNA keluar codon dekat ni, already exist in the A site, dia akan stop lah. Translation will stop. The others activated tRNA cannot bind to the A site already. So when it reach these either three of these codon, UAG, UAA, UGA. Paling penting kena hafal. Memang kena hafal. Tak boleh buat, tak boleh lari punya. UAG, UAA, UGA. Semua U punya daripada U punya. Semua U punya. So either UAG, UAA or UGA. Senang saja. Tak susah. Hanya 3 sahaja. So tambah kena hafal lah. Okay. Lepas tu, so since the stop already have a stop codon, then there will the release factor will bind to the A site. So this release, since the release factor already bind the A site, so other activated amino acid tRNA already cannot tak boleh masuk lah. Mereka tak boleh masuk lah. Because already it like act as the inhibitor lah. Dia macam release factor ni macam inhibitor lah. Already act as an inhibitor, dia sudah block the A site already. So yang ni sudah tak boleh masuk lah. Memang tak boleh masuk. So since the release factor already bind to the S site, so the protein shape like tRNA instead. Oh, okay. Okay, so this is the first step. Next, the step will also the tRNA at the P site that already contain peptide chain lah. Yang ni kami boleh panggil polypeptide chain lah. That already contain the final product. This is we can call this as the final product lah. Final product polypeptide chain. So the release factor will promote hydrolysis of the bond between tRNA in the P site. Means that dia ini akan hydrolyze the bond formation between tRNA and polypeptide lah. So it break down the bond between the tRNA and polypeptide and release the polypeptide and tRNA lah. And then final process is it require GTP and then 2 ribosomal unit where other component, all of these component large unit, small unit, and also mRNA will be hydrolyzed and released. Dia akan semua pecah lah. Dia akan semua not breakdown lah. It will separate lah. Tadi mereka combine together, yang ni combine together, and then they will separate lah. Separate, or we say dissociate. Kami panggil dissociate macam ni. All of the component in the ribosome, like large cell unit, small cell unit, release factor, and also mRNA will dissociate and release to the cell. Mana-mana sahaja, tak tahu ke mana lah. Ini maksudnya termination translation process is end. So this process is require GTP also. So finally your translation process is end. So this free polypeptide data it will be transferred to Golgi apparatus for the modification. Maybe dia akan pergi Golgi apparatus, dia akan pergi ke go to the Golgi apparatus and for the packaging and modification process. Packaging and modification process, and then to produce the protein that can be used in the cell, and then it will be released from the cell to the target cell by insulin, like the insulin or hemoglobin. Insulin will go to the pancreas cell, like hemoglobin it will go to the blood lah. Okay. So this is the overall of the translation process lah. So I think that's all for this live. So I just made a revision. So in this translation process, there are 4 main steps you have to know. First step is activation of tRNA, which is to form the tRNA that carry amino acid, just like that. And next step is initiation. This is the step that the mRNA then contains start codon. This is the start codon when it contains start codon, and this is the place that the translation will be start. Okay. Next is elongation. Elongation it requires ribosome unit, and for the polypeptide formation. polypeptide formation by adding amino acid from the activated amino acid DNA lah. Finally, termination is the process that to prevent the formation of polypeptide. And termination is reached when the mRNA contain these three codons UAG, UAA, and or UGA. Kalau ada salah satu tiga kolon ni, maksudnya translation sudah stop. Boleh stop lah. It already reached termination and will have the release factor, and this release factor will release the polypeptide and also dissociate all the component in ribosome and then this free polypeptide will go to the Golgi apparatus for further modification and packaging, and then it will be released through exocytosis lah. Okay, so these are the process of translation. So translation is much easier lah. I think lah, for me, translation is much easier compared to transcription. Because transcription is more complex and even it contain RNA processing, so it will much complex. So translation is more easier for me lah. But anyway, I hope that you guys can understand what I explained lah. So these are the feedback for. So hopefully, if you guys feel free, can scan this so I can know my performance lah. Nanti. Okay, sorry. So you can scan this feedback form lah. So good luck. I also take care. Have a nice day. Okay, thank you.